special interest groups on ensembles of conformations. And at the last CASP meeting, there was a discussion that we should create special interest groups to focus on some of the particularly challenging or important areas where modeling might play a significant role. And one of them is the idea of multiple conformational states. So a subgroup got together to organize this, and this is our vision statement: recognizing that biomolecules do not have static structures, but rather adopt a distribution of conformational states. The CASP SIG aims to evaluate if a critical assessment of predictions of conformational ensembles or multiple conformational states, say 2 states, if this is even feasible to do. And the SIG will consider and explore developments in the fields of structural and computational biology that are anticipated, and define protocols for assessing the accuracy of multiple or ensemble model predictions. And there was a little bit of this already in the CASP 15 and some progress. And John Molt gave a talk about what we learned in the first of these special interest group meetings. And if everyone could mute, and Cassie, if you can mute people who are not talking, that would be helpful. And when we talk about multiple conformationals, well, the typical thing is a protein may adopt 2 dominant conformations. A classic example is adenylate kinase. And can we somehow model the 2 alternative states and validate that some way? And at the other extreme is we typically call intrinsically disordered, I say it and fix my typo here, disordered proteins, where we might have tens or even hundreds of conformations in dynamic equilibrium. And so this is quite a challenging problem across this whole spectrum. And mostly we've been at the left side of the spectrum in our discussions, although as it go along, we are going to get involved in some discussions of modeling ensembles of disordered states. So this is the steering committee. And if anybody else is really interested in getting involved in this, we get regular advice from John, Christoph, and Andre. And really, the goal of this is it's actually, of course, to learn, but more than just to learn and explore ideas, it's to try to evolve. Can we actually do a CASP experiment, validating against some kind of data or structures? And I just want to mention and just advertise that we actually have 3 SIGs. The alternative conformational SIG, which is being held the first Wednesdays of each month at 11 Eastern Daylight Time. And we have another meeting in July, and then none in August and one in September. And then Arnie Ellefson and others are organizing a CASP AI SIG, really focusing broadly on how artificial intelligence methods and deep learning can impact structural biology and biology. And they put together a speaker group. There's been ongoing some very strong talks, particularly about methods and ideas. And I took this from their website, some upcoming talks. And now we've also started a CASP SIG focused on RNA. I think Rhiju and Rachel have been organizing this. The second one was yesterday, and this is on the first Tuesday of each month. So lots of opportunities to learn about deep learning and structural biology, courtesy of the CASP group. And I'm not going to go through this slide, we've presented it. So what's really critical in this multiple conformational state discussion is how can we make the models, but more, how can we validate the models? How can we have even a blind validation in the traditional CASP sense? And we've talked a little bit, Shoshona Wodak talked about a database of X-ray crystal structures of multiple conformational states. Other methods would include cryoEM and NMR. And we're going to hear a little bit about cryoEM as a method of looking at multiple conformational states today. And then there are other methods like cross-linking, even X-ray scattering and fluorescence studies. So this is the today's program. So that was my introduction. And then Hannah Wayment Steele from Brandeis is going to talk about some developments in multiple state modeling. And Rhiju Das from Stanford, who's been involved with CASP for some years, is going to talk about the particular problem of, or the feasibility of using cryoEM to characterize multiple conformational states of RNA. Rhiju, are you online now? I can't quite tell. Can you just say if you're there?
Hey, Guy, I'm here.
Okay, good, great. So actually, when we were organizing the meeting in Turkey, Hannah reached out to me and she asked if she could participate remotely, and we weren't able to set it up, but she had this strong interest already in last year. And about that time, or a little before that, she and her colleagues, including Dorothy Kern and Sergei Ovchinnikov, put a nice paper out in the bioarchives. And so the committee thought it would be great to hear Hannah's presentation and her ideas. And so, Hannah, can you try sharing your screen now? Looks good. So about 20 minutes, you can go a little longer and we'll save time for questions too.
Great. Well, thank you so much for that introduction, Guy. And yeah, I'm very excited to get to share this work with this group in particular. So as Guy mentioned, there's many other people who have been involved in this, and I just want to say a little bit before getting into the research itself. In particular, there's a fellow postdoc in the Kern lab, Dolapo Ojowo, who's been leading experimental efforts, particularly NMR, to test a number of predictions that we made in our original preprint. And so working in collaboration with her has just been a really great experience. Julia Apitz, who I believe is also here, is a new PhD student in the lab who has also made really critical contributions to this. And I've been very fortunate to receive advice more on the theory and modeling side from Sergei Ovchinnikov, from Lucy Caldwell at Google, and of course from my advisor, Dorothy Kern. So as Guy laid out really nicely, we are now poised where we have tools like AlphaFold that we know can make very accurate predictions for single structures. And when I first met Doro, when we started talking, we really wanted to see if it's possible to use deep learning, possibly in architectures like AlphaFold or others, but to try to predict multiple conformations of proteins. And I'm showing this figure from this review to try to highlight the point that there's many timescales of motions, and understanding these is critical to get from just being able to predict single structures to more completely understand proteins and other biomolecules in the context of biology. And so we have this idea that AlphaFold is a very good deep learning architecture that can go from evolutionary information at the sequence level to these atomic level accuracy predictions. And so then when we were thinking about, can we, for the problem of multiple conformations, when we're thinking about trying to use that information of evolutionary coupling, if we can use that to predict multiple conformations, we thought that it would be best to try to start with the largest scale conformational changes that we can find, thinking there would be the most signal for that. And so if we could see anything, hopefully it would be these largest scale changes. And so a set of proteins that fit the bill for that are these so-called metamorphic proteins. So metamorphic proteins are defined as proteins that switch their secondary structure as part of their biological function. A well-studied example of this is the RFAH protein from E. coli. And it's really interesting because it actually switches between being a transcription factor and a translation factor. Here, so these are the two known states of it. And you can see that this C-terminal domain highlighted in red between one state is in a helical bundle, and then upon binding another partner, switches to this more beta strand form. So the ones that are known range from simple organisms like E. coli to more complicated organisms like humans. So this is another example of two known states with different secondary structures from the MAD2 protein, which functions as a cell cycle checkpoint. And I'm very interested in these proteins because they totally challenge the notion of a single sequence going to a single structure, even then going to a single function. And when we came to this problem, Lauren Porter had done a very good, very thorough job of demonstrating that AlphaFold in its default settings fails to predict more than one of these conformations for a set of known fold switchers. But we didn't want to give up on AlphaFold because there was a really clear contradiction that it gave for a fold switcher that has been studied for a long time in the Kern lab. So CAIB is a small protein. It's only about 100 residues long, but in the course of its function, it has this really dramatic change in its secondary structure. So you might be familiar with CAIB because it's part of a classic oscillator in biology. So it, along with Chi A and Chi C, regulate circadian rhythms in cyanobacteria. And I won't go through the whole mechanism of how that works, but a core part of that is this fold switch. So by day, CAIB exists in this so-called ground state, where the C-terminal domain is making this helix strand structure. And at night, Chi-C autophosphorylates and forms a binding component state that drives the equilibrium of CAIB then to this fold switched state. And the structure of that is very different. So the C-terminus is now in this beta-beta helix conformation. And it's interesting to note, it goes from this more topologically knotted structure to then the beta strand has to come out and come to the outside of the protein. When we take this protein and perform NMR on it, we see that both structures are present at equilibrium, with the fold switch state being populated to about 10%. So it's the more unfavored state. But the collab fold prediction, and members of the lab had tried a number of things, always gave us back this more thermodynamically unstable fold switched state. So there's a contradiction between whatever coevolutionary information AlphaFold is picking up on and what we actually know thermodynamically to be more stable. So when I joined the lab, I was trying a number of things, and something actually really simple ended up giving me a prediction of the ground state. And that was if I took from the MSA that CollabFold generates, if I took the closest 50 sequences by edit distance and just used those as an MSA, it gave back the ground state. But then when I kept playing with things, if I took the closest 100 sequences, so not that big a difference in the scheme of how many sequences there were, then it switched back to the fold switched state. So we hypothesized that within that MSA returned by CollabFold, there were pockets of signal for both of these states. And so I'm just going to show a schematic, but I'll get more into details of the actual clustering algorithm that we converged on in a couple slides. But the idea is that if we take a MSA and cluster it in a very heuristic agnostic way, and then use those clusters as input MSAs, we thought then that we would get a distribution of different structure predictions back. So these are all samples, models for CAIB. And so if we did that for CAIB and we plotted those samples that we got back and compared them to these two known structures for CAIB, it was really interesting to see that there were two regions of this distribution that the AlphaFold intrinsic confidence measure, PLDDT, where that was higher. And those regions corresponded to what we knew to be the two main states of this protein, the ground state and the fold switched state. We wanted to compare this to another method for MSA subsampling that had been published at the time. So Jens Meiler's group demonstrated that if they took an MSA and randomly sampled sequences from it, sort of another way of imparting noise into AlphaFold, that they could sample multiple states of ion channels. Well, we found that failed to return the same quality of structures for the ground state. And for larger MSAs, that uniform sampling returned no structures for the ground state. And this was not unique to AlphaFold being a supervised learning thing. We tried, in particular, other unsupervised learning methods and found, so this is showing for the method MSA transformer, that if we took those same clusters and put them in MSA transformer, within them we also saw signals corresponding to the two different states of CAIB. And after our initial preprint, Lauren Porter went and did a much more thorough demonstration as well, that using MSA transformer and others, they could see signal for multiple states. So the first question we wanted to dig into with this observation is, so why is there this signal for two states? So that fold switch state is a thyroidoxin-like state. And there's a lot of thyroidoxin-like states in the PDB and just proteins in general. So we were curious if there was signal for that fold switch state from things which we knew to the best of our ability were actually CAI-B. And so we took a CAI-B phylogenetic tree that a former grad student in the lab had developed. So these are things to the best of our ability we know are CAI-Bs. And we performed AlphaFold predictions for each of them. And as a MSA, used just the 10 closest sequences of things that are truly CAI-B. And so this is showing that phylogenetic tree and coloring each sequence by whether it predicted the ground state or the fold switch state. So it was actually a bit surprising to us. There were CAI-B variants that were predicted to fold into the fold switch state. And if we look at that same tree colored by the PLDDT, they actually had high confidence for that fold switch state. So we went through and tried to map these predictions to what we know in the literature about CAI-B variants that have been characterized. So starting with the oldest variants, the ones that have been used as models for understanding circadian rhythms from cyanobacteria, these folded to the ground state, although interestingly with lower confidence than other parts of the tree. So those match our expectations for what would be their thermodynamically most stable state. The Kern lab recently discovered an even more primitive CAI system for regulating circadian rhythms, which also has a CAI-B that undergoes a fold switch, and that also predicted the ground state. So that matches our expectation. Trying to look elsewhere in the tree, there was a crystal structure of a CAI-B variant, which this group demonstrated was not involved in circadian rhythms and is actually more likely involved in a stress response and is stable in this fold switch state. So it turns out it's really great if you actually read the literature, and you can learn a lot. So we wanted to test some of our other predictions from, there are about 500 variants in this. So we selected another copy of CAI-B in one of these model cyanobacteria that has been, where the other variant has been used to understand the circadian rhythms, where it has a high confidence for the fold switch state. And so Delapo and Julia, being mentored by Delapo in a rotation project, performed NMR and characterized this, and the results are quite exciting. And I think it might have been one of the most successful rotation projects I've ever seen. So another line of questioning we had at the same time is, what is the sensitivity of AlphaFold to predicting both of these states? Something that we observed right away is that when we looked at these different parts of the MSA that were predicting one state or the other state, if we looked at the differences in sequence positions at those two locations, we noticed that there were quite a few locations where the residues that were enriched for predicting one state or the other state were actually quite different in their chemical content and hydrophobicity. And so we were curious if this is sort of just in a brute force way, because there's a lot of ways to design proteins now, but is there any way that we could just select a number of these minimal mutations that actually would be sufficient to cause a CAI-B to switch its fold? And so a necessary first step to designing that would be, can AlphaFold actually predict with high confidence the effect of a small number of mutations? And so Delapo and I collaborated on this, and the design scheme that we ended up on is we used our CAI-B variant, which has been studied in the lab. We started with that one, and then just using AlphaFold as a folding engine, folded all variants of these most enriched mutations, which were stabilized for the other state, for the fold switch state. And we found that as few as 3 mutations were sufficient to make a prediction with a high confidence for the other state. And the results when we test this mutant in NMR are, I'm afraid I don't have them to share here, but they're quite exciting. So that was a bit of our dive into why and what's the extent of how that method can work on Chi-B. But I wanted to create a method that could be generalizable, that could, if it were possible to use on other proteins, didn't require deep knowledge about the particular phylogeny or evolution of any particular protein family. But if you've ever worked with clustering algorithms, you know that they have a lot of tunable parameters, and it's sort of annoying to work with in that sense. So I ended up using a method based on the DB scan algorithm, which at its core has one parameter, which is basically the distance between points at which it decides that 2 points should be in a cluster or not. And I found that if I varied this epsilon parameter clustering a given MSA, that there was a value of that epsilon for which there would be a maximum number of clusters. So this t-SNE plot is demonstrating what clustering would look like at different values of that. So at a lower epsilon, DB scan doesn't cluster all the points, it throws some out as noise. And so below that, there are fewer clusters because it's throwing out a lot of noise, but over that value, it's then lumping clusters together. So there's no theoretical reason for why this should be the best method, but empirically, we reason that this might be a way to maximize the content, the information content of our clustering. So when we used that to go and try this method on other proteins, it was then really satisfying to see that we could get back AlphaFold models for these different other fold switching proteins. And that I was really curious to then try to use this method, and it has limitations, which I'm happy to talk more about later. But even just this first naive heuristic approach, could we use this to try to find new fold switchers? And just as a back of the envelope calculation, so in the AlphaFold database, there's roughly 200 million protein structure predictions. Again, from great work from Lauren Porter, at one point, she just went through the existing PDB and tallied all the structures that have different secondary structures, and through this way developed her benchmark of proteins that are computationally identified as fold switchers from within the PDB. So if we just take that number at face value, that would mean that there's roughly 1 to 8 million fold switching proteins that, by only predicting a single structure, we're missing out on that heterogeneity and a ton potentially to learn from all of those. So as a really rough, just first pass at this, we took about 600 proteins from a formerly developed data set from the Baker lab. We limited them to proteins only up to 150 residues in length and ones that had a lot of MSAs of sequences in the MSA. So we performed our clustering and then only were sampling 10 clusters for each. But so this is a plot of those results. And each gray trace is one of these proteins that we sampled. The x-axis is showing the change in RMSD from each sample to the known crystal structure. And the y-axis is then the AlphaFold confidence. And so you see that for the majority of them, as you get farther away from the known crystal structure, the confidence goes down. And so that it was expected for a protein for which there wouldn't be, where there's not strong signal for something else. But there were a few traces where there was a high confidence for a structure that was far away from the known crystal structure. And so this red trace, we then, for a few of them, went and performed more extensive sampling. And so this is then just a PCA plot of the more full landscape, and showing that there's one peak which has high confidence, which corresponds to the crystal structure, and then this high confidence alternate state. And so this protein turns out to be a secreted protein by tuberculosis. Little is known about it, but I'll just highlight the difference between the known structure and this alternate state. And again, we're currently performing NMR to try to tease out, does this other structure exist and to what population? But so on the left is the known structure, and you can see that there's this yellow strand in the middle of this, the beta 5. In the alternate state, it gets replaced by the beta 1 strand. Another change is that this alpha-4 helix swings from one side to the other side of the protein. And very interestingly, this is also a thioredoxin, like the fold switch state of Kaibi. So in summary, we found that just by simply clustering sequences, we could use AlphaFold to predict states of multiple conformations for known metamorphic proteins. And this is also now allowing us to test predictions of new alternate states for other proteins. And our rationale as to why this method works is that these families of metamorphic proteins contain these pockets that are stabilized for both states. So there's a lot of directions for that I'm really excited about with this work. On the biology side, I just continue to be fascinated by metamorphic proteins, and now with tools like this AF cluster in hand, we can really try to ask more questions. How do they evolve? Why does nature select that type of mechanism? And ultimately, can we find rules governing what sorts of fold switches might be possible? On the modeling side, though, which perhaps is maybe of more interest here, I'm showing here, so there are, as I'm sure you appreciate, there's a lot more complicated dynamics out there than only these changes between these very extreme conformational states. A protein that people think a lot about in the Kern lab are kinases. And so at one point I tried this AF cluster method because there were some kinase targets in the last round of CASP. And we did find, for instance, this is for the BRAF kinase, that we got models that recapitulated both an active state and an inactive state. So those are the two main known alternate states of kinases. But in the models that I was looking at, there were a lot of just other conformations. And when we do NMR on kinases, we know that there's more than just these two states present. And so this highlighted to me the importance that if we want to get better at predicting dynamics of multiple timescales and multiple types, we need better quantitative benchmarks for what those dynamics are. So that is where I've been spending my time, and I'm really excited about that work, and also the many other directions that we've heard about in this group so far. I believe that's all I have. And again, I want to acknowledge the folks who have made this possible. I've had very great discussions with other members of the Kern lab, as well as Lucy's team at Google Brain, and I'm glad to answer questions.
Yeah. Well, thank you very much, Hannah. It's very exciting, very nice work. And I want to move on, and there's no questions in the chat yet on the talk, but I will allow one person who just wants to jump in and ask their question. If somebody wants to just jump in and ask their question, then I'm going to move on to the next presentation.
I just have a very quick question. It's Arne here, and could you detect your fold switches with the MSA transformer or something like that? Because if you could do that, you could do it a much faster screen.
That's a great point. Yeah, we can. I like that AlphaFold gives you real 3D models, but you're correct in that if you want to go and screen the whole proteome, that something like MSA transformer would get you there faster.
Okay, I'm going to move on to the next talk in the interest of time, and we did schedule time at the end for more discussion, and we can come back to Hannah's presentation. So Rhiju is a structural biologist, and particularly high impact work in cryoEM, and has been participating in our CASP program for some time. Rhiju is at Stanford University. So Rhiju, well, just go ahead.
Thank you. Just before I start, I want to acknowledge that many of these ideas and slides are from a PhD student here at Stanford, Rachel Kresh. Some of you know her. She'll be speaking later in the Ensemble SIG later this year. So think of this as a preview of her work, which I think is really impressive. And so, Hannah just told us about these fold switching proteins, which appear to, there appear to be lots of them, although maybe not so much. Maybe like a few % of proteins might have that fold switching ability. There's something that we see often in the RNA field, and this is a quote from Rachel Kresh from one of her slides from when she was co-assessing the CASP RNA category, where she said, truly the majority of our RNA targets can be seen as ensembles, and RNA may end up, hence RNA may be a key category in ensemble prediction. So the majority of the 12 RNA targets in CASP15 showed evidence of having multiple states. Some of that came from crystallography, where the copies and asymmetric units didn't quite match up, but a lot of it came from cryo-EM. And I would say prior to maybe 2022, 2021, we weren't quite sure if these multiple states that were being captured by cryo-EM were totally real. Maybe they were just due to noise that's being picked up in data processing algorithms. But I think many of us now feel much more comfortable in saying that we're picking up legit conformational states. And for me, the really important results started to happen last summer that really convinced me that RNA cryo-EM will be a gold mine of multi-state ensembles and potentially really great CASP targets for future ensemble modeling. So what I'm showing here are structures of a molecule called the tetrahymidoribozyme. This was the first RNA-only enzyme discovered by Tom Cech back in the early 80s. It won a Nobel Prize, but its structure hadn't been solved until 2021. This is work by my lab and Hua-Chu's lab. And on the left, I'm showing you a structure of this ribozyme at about 3 angstroms resolution. And we knew that we were right because on the same day that we published our structure, Di Lu, who was in Joe Piccirilli and Pong Yin's lab, put out a structure in bioRNA that was basically indistinguishable at atomic resolution. What convinced me then a few months later that we were also capturing multiple states accurately by cryo-EM is a structure here on the left. Can you see my pointer? Can folks see the pointer?
Yeah.
Okay, good. Then, so it's the structure here, sorry, on the right is a misfolded state of this RNA. So this RNA, if you just add magnesium to it in vitro and try to get it to fold, at room temperature, it actually, the majority of it folds into an incorrect non-functional state. It's been called a misfolded state. And on cryo-EM, on data taken on the same grids as with the native state, we can see this alternative conformation if we intentionally don't let it misfold. And so the structure that we ended up with and published in 2022 is shown in green. And then in brown is a structure that Jeff Keefe's lab, so this is Steve Vanilla and Jeff Keefe's lab, had independently solved. And they overlay at nearly atomic, with nearly atomic precision over most of the RNA. And there's actually a shift in the catalytic core here, which actually explains, there's a tangle in the RNA fold, which likely explains why this RNA is misfolding and why it sticks around for so long as a misfolded state. I just want to emphasize that this is a neat kind of category of multiple conformations that's a little bit different from the fold switching of proteins that Anna talked about. These are, they're literally conformations solved for the same RNA on the same grids and automatically pulled out from standard Reliant classification software for cryo-EM. We believe it again because there's atomic resolution agreement between independent groups, and we also believe that in this case where we reached near atomic accuracy, these kinds of folds will be, and alternative folds will be well-defined targets for prediction in future CASPs. I do want to emphasize something that Guy also pointed out in his introduction. These are not Boltzmann ensembles, not just because they've been flash frozen, but also because intentionally these RNAs have been folded out of equilibrium, and these this misfolded state, it lasts for days, but if you could wait for years, or if you heat up the sample, it'll eventually go to the native state on the left. But these are still really interesting. And so in this case, this alternative state, I'm not going to go to details, but this misfolded state has a topological tangle that we believe is likely to be happening in other RNA systems, including parts of the SARS coronavirus 2 virus and the ribosome, as they fold or misfold during their biological life cycles. So this is a case where we have discrete states, multiple states. And the good news is that I think, for CASP16, we'll be able to include these if they arise as targets. We'll be able to include these as CASP targets because CASP basically knows how to handle modeling problems with discrete heterogeneity. So John talked about this in the last ensemble SIG, but I just want to highlight for proteins where there were challenges to predict multiple conformations of proteins. We had an example of that also in this last CASP for RNA. This is target R1138. This is solved by Ewan McCrae, who's here on the call. working with Ebby Anderson. They had solved the structure of an RNA nanostructure they designed, and on the right-hand side is the so-called mature conformation, kind of the final probably equilibrium structure of the RNA. But on the left is what they dubbed a young conformation of the RNA. This is what happens after, I guess, 15 or minutes or 30 minutes after the RNA is transcribed and then tries to fold in its own. It gets stuck in a kinetic trap. This actually also interestingly has a topological tangle that prevents it from getting to the mature state. And in CASP, Andre put out the target with additional information saying, this is the co-transcriptional product, the structure observed in cryo-EM grids immediately after transcription, and then around 8 hours later have alternative conformations. I think the one mistake that we made in presenting this target was we said you can submit alternative conformations as separate models within the 5 that you proposed, but I think in retrospect, it would have been a good idea to have 2 separate targets, R1138, R1139, where we explicitly say, actually give us something like the young conformation or the mature conformation with some sort of description. So I do think that in the future, this is what we're going to, in Cas16, we can have more of these multiple conformation targets, and RNA will be probably enriched. When we talk to RNA and RNA protein experimentalists, if we just ask them if they've been able to solve multiple discrete conformations of their complexes, I think that'll elicit from these groups, hey, actually, we had this 30% or 20% of our particles went into a different class. Why don't we try to make that a second CASP target? I do want to say that I wish that we'd made these discrete states, make each of these states a different CASP16 target or a CASP15 target, and we should do that in the future. And then those targets can be, when we provide a description, we should, of course, tell modelers how those states were separated or trapped. For cases where there were ligands that trapped one conformation versus the other, or mutations, we can specify that. We've already been doing that for proteins. For kinetic traps, I think we should specify the RNA preparation history, which is what Andre did, I think, with advice from you and Evie in the last CASP team. So we should state that. One thing that's interesting that I don't think has happened in CASP before is, what if instead of maybe the 2 RNA states arose from the same preparation history for the same sequence, without ligands, but we see them, they get separated automatically during cryo-EM classification. I'm not sure how you then present them as separate targets. Maybe we can ask the experimentalists who are experts in the system provide some sort of natural language description of how the states differ. And that'll be interesting from the point of view of automated servers who are trying to make the predictions, because then they'll have to be able to understand English language and read the descriptions and take that into account when making their predictions. That maybe is the one twist I can see going forward in the next CASPs. Okay, so that's discrete heterogeneity, multiple states, very much along the lines of what Hannah talked about, or what Guy alluded to at the beginning of this session. I think there's something more interesting, though, that we can do in CASP16, which is to look at continuous heterogeneity. So, I think many of us have seen these MD simulations of molecules, and we're struck by how much motion is predicted in these simulations. This is the simulation of that tetrahydroribozyme, again here from Rachel Kresh with advice from David Case. And I've never known whether to kind of believe these fluctuations or not. So kind of the base pairings in the RNA are the same. At a local level, the RNA structure is staying intact. The ions are moving around in the water, but you can see the helices are also flopping around. So is that real or not? And I think there's an opportunity now with cryo-EM to potentially evaluate different MD algorithms for whether they're predicting or mispredicting these kinds of fluctuations. And so, and the reason for thinking that is because I think for the majority of RNAs that we and others have looked at, we see evidence for these continuous fluctuations from cryo-EM using algorithms like 3D variability analysis from CryoSpark. So here on the left is a movie from Ewan's paper on that R1138 nanostructure, where he's showing what look like principal components of motion that are inferred by 3D by looking at the individual particle stacks. And these motions, they look physically realistic. Like the bends here are happening at a kissing loop in the RNA rather than in the helices themselves. So these they look kind of legitimate. On the top right here is another CASP target where that Rachel solved. It's a part of a coronavirus RNA. And it, again, 3DVA automatically seems to capture this bending motion of a helix. On the bottom right is actually that same molecule I showed you before that we solved in 2 different structures at near atomic resolution. The misfolded structure, actually with enough particles, Reliance subclassifies that into 3 different states, which we call the M1, M2, and M3 here. But these look like they're just kind of arbitrary subclassifications of some sort of continuous dynamics or heterogeneity. Okay, and then we have additional evidence, which we haven't published yet, that even if you look at those tetrahedral ribosome data sets, even at the ones that are atomically defined, with some of the newer algorithms coming out, like cryoDRAGON or E2GMM, that look at the individual particle stacks, there's additional motions that we see even outside this core region that are just present in the RNA. So I think you just get these conformational fluctuations sort of for free with RNA, and they're really interesting. And they're just sitting there in the cryoEM data. So can we do something about that? So I think this could be super exciting. So instead of using something like GDT on your RNA conformation or on your 2 RNA conformations, what if we in Cas16 for a subset of targets we accepted not 5 single structure models, but 5 ensembles with, say, like 100 models in each of them, or 1,000 models, I don't know, and optionally 100 weights for each of those coordinate models. And then we could evaluate based on a direct comparison to the cryoEM particle micrographs. And so there's a nice framework for this that was proposed by Pilar Cosio and Gerhard Hummer, called BioEM. And just in the last year or 2, they've shown how to dramatically accelerate calculations to get a real likelihood function. Or actually better than that, you get a log posterior. So for every model that would be submitted to a CASP, or actually every ensemble of models, by direct comparison against the particle micrographs, you could get out a number, which is the posterior probability of the model given the data. There's going to be an issue in terms of storage and compute, because we're going to have to now store 100x or 1000x more models. But if it's for a subset of targets, that doesn't sound that bad. And then there's going to be GPU compute required, I think, to then score hundreds of multi-ensemble models. But it seems like, I don't know, it seems manageable. There's certainly proof of concept out there. And Rachel will tell you maybe more about this later in the year. And then it would be great to draw assessors from the EM community, or even folks like Pilar. This actually could work not just for RNA. I think RNA will provide some of the really interesting test cases, but for folks solving protein and ribonucleotide protein assemblies like the ribosome or the spliceosome or telomerase, they have these fluctuations too in their data. And I think what's cool about this is that you could guarantee that there are CASP targets, CASP16 targets, because you could ask folks for, actually, you could ask cryoEM microscopists for data sets where they've actually already published the structure, because they've typically only used 20% of their, or 30% of their particles to produce their high resolution, atomic resolution structure. And if they haven't yet released their raw micrographs, then you could actually tell CASP modelers, hey, we think we have the dominant single structure. It's in the PDB already, but now predict the fluctuations around the structure using AMBER or CHARM or coarse grain MD simulations. And then they would be scored against these previously unreleased micrographs that all the different that my group and Wild's group and all the different groups have sitting on disk. So you could guarantee having 12 ensemble modeling targets coming from RNA, RNA protein, and probably protein assemblies as well. So that's the proposal. I just want to end by describing the precedent for how to assess ensembles, again, coming from the RNA world, although mainly at the level of secondary structure. And I stole this slide from Hannah. Hannah, you're acknowledging the next slide. So our group has taken part, or has helped organize a kind of a set of assessments for modeling methods that predict RNA secondary structure. So if you have a bunch of RNAs and you think that they fold up into, maybe some of them fold up into multiple structures, there's this technique that we can do pretty easily and pretty cheaply, where we provide a chemical modifier that tags the nucleotides in the RNA that are unpaired. Okay, and there's several of these. And then there's all sorts of tricks that we and others have developed that let us then figure out, using deep sequencing, which parts of the RNAs appear to be unpaired, which ones are chemically modified. And we can take these raw data and compare them to the predictions of the secondary structure ensembles for existing algorithms. And so Hannah showed in a paper that we published at the end of last year. So I should probably mention, so Hannah did her PhD at Stanford also, and then I had the good fortune of working with her then, and she worked in RNA before. And so from these data, this is kind of a CASP style Z-score heat map, where we could rank different methods that are out there for predicting ensembles. So this was, and we got and actually these data, we initially looked at chemical mapping data that our lab had collected, but our final assessment here was based on lab data collected from, I think, 10 or 11 different labs around the world. So this kind of like, these are secondary structural ensemble methods, but I think it's now time to actually test the modeling of tertiary structural ensembles, and I think RNA cryo-EM is going to be the useful observable here. All right, so okay, I'm going to skip that. All right, so summary here. There's kind of two flavors of heterogeneity or flexibility that we see in macromolecules. They're both present for RNAs. For discrete heterogeneity, I think the key thing here is that when we start recruiting experimentalists like Ewan and others and ask them, do you have targets for CASP16? Let's just ask if they have any that they've solved in multiple discrete states and present them as separate targets. In terms of continuous heterogeneity, that I don't think has been assessed in CASP before, but it's just, I think it's there in all macromolecules. And for RNA, there are these really interesting dynamic motions. And there's a prospect here of assessing models coming from, let's say, MD simulations by direct comparison to the cryo-EM particle stacks. So I'm advocating that we think about that kind of a category for the next CASP. And it would have a big impact because I know at least for RNA MD, I don't know which force field to use. I don't know which water models to use. I don't even know if the all-atom models, if the range of fluctuations makes sense or where there's motion. And so just in terms of ranking models to use, it would be a huge, I think, a huge gain for the computational biophysics field. All right, and then I just also want to end, this is a short talk, but I want to make suggestions for upcoming speakers and also make sure that everyone sends guide notes for additional speakers. I've already mentioned Pilar, who I don't know personally, but I'm a big admirer of her work for advice on BioEM. I think she'd be a great speaker. Steve Bonilla is setting up his own group at Rochester, and he's doing a lot of work on getting ensembles of RNAs and ribonucleoproteins proteins with cryo-EM. Rachel, I've already mentioned. Ellen Zhang and Wayment Steele are developing these algorithmic approaches to take the cryo-EM data and infer ensembles, and those could provide basically kind of best case scenario. What is the best you could possibly get for an ensemble model in terms of matching the cryo-EM data. And then I think it would be good to, if you think this is a good idea to assess continuous fluctuations, we might want to go and now bring in folks coming who are doing MD simulation. David Case is someone who could both give a talk and also probably propose additional speakers who are doing MD, not just in RNA, but ribonucleoproteins and proteins as well. That's all I have. I want to thank Rachel, Philip, and Ramya, who are co-assessors in CASP with me, all of these folks who have helped us with the CASP RNA assessment, and our funders.
Well, that was great, Rhiju. Very, very.
CASP with me, all of these folks who have helped us with the CASP RNA assessment, and our funders.
Well, that was great, Rhiju. Very, very helpful. I'm gonna go to this and just point out one just administrative thing. I noticed that our participants keep topping out at 100, and we actually had modified our Zoom for more than 100, but I think maybe it had to be updated. So a number of people weren't able to get on, and in any case, if they sign up on the mailing list, the videos will be available there and they could hear it. And I appreciate Rhiju's suggestions for speakers, and if other people have other suggestions, we do have a committee that we will evaluate, to just prioritize those suggestions. And that was very helpful, Rhiju. So we have a little bit of time left, and I thought we could spend a few minutes with a few more questions to the speakers, and then try to get a little bit more into how to make this work for CASP. And I actually want to maybe take because I'm the MC, to ask my question first, which is actually to Rhiju. And the question is this, is that if you had a bunch of incorrect models and then you just look at the morph between them, they'll look like beautiful dynamics, but it's not dynamics, it's just incorrect. And so the question is, how can we be rigorous with cryo-EM, and even particularly RNA cryo-EM, but it's similar with proteins, that we really do have two states and we're not just overfitting the data, both discrete states or continuous states? Is this something that is rigorous from the analysis of the cryo-EM data, or how can we be sure of that?
So I think the detailed answer to your question will be what Rachel talks about later in the year. I think there's still, I have to say, there's the only reason that I believe the alternative states is because now, at least for one molecule, we have multiple independent groups who have gotten the same answer.
Yeah,
With, different preparation, slightly different preparations, different instrumentation, different data processing, slightly different data processing. But there's still this possibility of Einstein from noise.
Dependence, right? You see it changes with time and you feel reliable. But when you have one data set and you pull out 2 or 3 or 8 conformers, that's what we'd love to do, right? But I'm just wondering, are the metrics, are the quality assessment metrics sufficient to be able to conclude that they're really independent states and not just errors in the modeling?
Yeah, so I think there is this worry that there's an Einstein from noise effect, that that the data processing with the cryo-EM is pulling out states that aren't really there, whether they're two discrete states, and then even worse when there are these fluctuations. So what Rachel and I and Wa have been talking about is what we might need is something like tilt pairs. So this is an idea that was kind of proposed about 10, 12 years ago for cryo-EM, just single structure solving. That if you have a model like, oh, I have an RNA with 2 states, one way to rigorously test whether you have those 2 states is to collect independent data that are kept hidden from the analysis. And one way to do that is to take, for the same grids, micrographs of all the particles, and then you tilt the grid and you take micrographs of the same particles again. If your models are legit, like my 2-state model, or even my conformational ensemble, continuous conformational ensemble, you should then be able to predict within experimental error what is the micrograph going to look like when you tilt the grid. Yeah, so I think that's maybe the one thing that we need to do.
Another thing is cross-validation, where you just omit part of the data and then see if you validate your models when you test against the part that you omit. And I guess you can do that with the cryo-EM reconstructions.
We can do that a bit with the cryo-EM reconstructions. It's still like, I would be more convinced. I think Rachel and I would be more convinced if you have the tilt pair data, at least for some paradigmatic examples. And then maybe some sort of internal cross-validation equivalent to an R3 or something could be used for, you do the tilt pair on like 10 data sets or 10 molecules, and then perhaps you would validate the validation criterion for something like an R3 internal cross-validation for every other data set.
So there are a bunch of questions in the chat, and we won't get to all of them because I want a little more time to talk. And I would appreciate if Rhiju and Hannah could actually try to answer some of these questions, and we'll keep the line open a little bit longer than the 12 o'clock. But I guess I would just choose one more of these related to. I'm going to let somebody just jump in. Maybe one more related to Rhiju's presentation. Does somebody else want to ask something? Just jump right in. Arnie, go ahead.
Okay, nobody else thought. RNA is great, but I think the most people have a protein interest, and I think it's MD simulations of RNA is much harder for proteins. So I wonder if you take single chain proteins in cryo-EM data, would you see enough fluctuations to be interesting in a type of protein to validate MD simulations? Or is it really because that RNA is more flexible and bigger that you are able to see that? Because sizes also matter, I guess.
So we haven't personally checked, but it is striking that when Ellen Zhang or Wayment Chen, when they are developing the algorithms, they typically they'll often use a ribosome data set or a spliceosome data set, which are, I guess the spliceosome is mostly proteins, although has an RNA machine at its core.
I mean, they're very big.
There are some characteristic protein data sets.
Yeah, I mean, if you take like I mean, 500 recipe proteins, you have 5 kilodalton protein. It's hard to pick it's hard to pick it, and I guess we need like 500,000 picks to be able to see anything really, because like it's. I mean, I think it would be easy for big things in general to have the fluctuation.
I think it'll be easier for big things. Yeah, I agree with you. Yeah, and I think those data sets are available. Yeah, it would also be interesting to have as a target a protein, like even just like one, like, well, apoferritin would be a problem, but a protein where maybe we don't expect there to be a lot of fluctuations. And then MD simulations of those proteins, you know, actually to do a good job, they have to not predict crazy fluctuations. So I think, you know, including proteins, even if they don't have a lot of dynamics, backbone dynamics, could still be very useful.
Okay, I want to move to these questions for discussion that I have here, spend a few minutes. And actually what I want to do is allow people to continue some discussion after the end of the hour, kind of informally on the line. I'll keep it open if the speakers don't mind to stick around for a few more minutes to chat. But some of the other ideas I wanted to look at in this session today is, so the last 3 sessions, including today, mostly have focused on this kind of 2 alternative state modeling. And Rhiju actually got into this idea of continuous motions. And that at some level relates also to the idea of intrinsically disordered states, where you have a wide distribution of conformations. And one of the things the executive committee, or the organizing committee, I should say, of the SIG has been discussing is this really, should this really be part of a CASP 16? Is it feasible to generate data or models that you could validate predictions against it? And probably it would be data. And I think it actually is very much like what Rhiju talked about with the continuous states, but continuous states, you know, with different populations of them among disordered states of biomolecules. So does anybody have any thoughts or views or advice to us about this? Should we include this? I'll use raised hands for now. I saw Kumaran raised his hand. Why don't you say something, Kumaran?
Yeah, so I work for the PDB, so we have this issue for a long time. So one of the issues is we need to have a clear definitions of what is the state versus the state is not an equilibrium distribution. A model can be a different state, also can be a part of the equilibrium distribution. How do we distinguish these two? We leave it to the users to decide. The person who solves the structures knows these are the 2 distinct states, or is there a more generic definition? Okay, you have this much RMST between an equilibrium distribution, so we can say these are the 2 different states. So what are the definitions here?
Mm-hmm. Well, it's going to become an issue as you assess too, that way. Does someone else have any more thoughts about whether this, whether CASP 16 should include continuous or extensive distributions of conformations rather than the kind of things that Hannah was talking about?
Guy, could I ask Rhiju a question?
Please, go ahead.
I'm curious if for these conformational motions, if there's any systems for which people have a really good idea as to the time scale of those motions.
I mean, Guy should answer that. Or the other NMR spectroscopists.
Well, I guess if you're seeing them in the cryo-EM, it says they give some sort of boundary on how fast they have to interconvert. I don't know the time scale, but there's some sort of limit where if it interconverts much faster than that, you're just going to get an average, just like an average chemical shift, just like fast exchange. there must be something like that in the cryo-EM experiment, but these are frozen out on the time frame of they're frozen out in the cryo-EM experiment itself. But at room temperature or in a non-frozen state, they may interconvert much faster.
I guess-
Yeah, I can add to that quickly with numbers. Simulation data of like the freezing process says that boundaries of around 10 kilojoules are like no longer visible after freezing at the timescales we're freezing. So that's the type of barriers, at least simulation says, we're freezing out.
I want to go to these last 4 questions and deal with them just kind of all together. And probably these discussions should go longer. Can we use cryo-EM data in CASP 16? And I think Rhiju is making a strong case that we can. And this would be a kind of data for alternate conformations. What other kinds of data or experiments should we be talking about in this group as ground truth data for a CASP 16 experiment? And one point I will make is we are doing something progress here. It may seem like we're just running against this wall, but as Rhiju pointed out, by having these discussions and by reaching out to the community and involving the community in the discussions, we're making them aware that this is a CASP 16 project. So hopefully they will identify from their own research labs test cases for us to use. So propagating this concept across the community hopefully will increase our set of targets. But I'll just ask these questions then. Is there a particular strength of cryo-EM that it really should be our focus, or are there other types of data that we should be bringing into this experiment?
I mean, I'll speak out again. I think NMR is the place for which for proteins of single domain size, where we can really differentiate between what are fast motions and what are slow motions. It's a continuum between, I guess, the continuum of fast things and more discrete states. But I know that makes for a lot harder evaluation.
Well, size is generally a challenge there, but you're right, NMR is particularly.
But I mean, it's kind of there's 2 different size challenges, because as we've said, I'm actually not familiar what the smallest protein and that one can get data for in cryo. But to me, they seem like complementary challenges.
Yeah, I think they are complementary techniques today, because NMR is limited in upper size and cryo-EM to some degree limited on the lower size. And there may be some things that could be studied by both. I think FRET data, a solid state NMR has the potential to isolate states, and of course, crystallography.
Can I ask Hannah? So, I mean, you say NMR, but what more exact do you want to measure?
Well, from the NMR, you can model a simple example as you have a cyclopeptide with cis-trans isomerism, and that results in 2 conformational ensembles, one with cis, one with trans. That's very simple thinking, but the same is true even for 200, 300 residue proteins. Adenylate kinase is an example where there's
so you see alternative NOEs basically, or
alternative NOEs, or it could be slow exchange, so you have a complete set of peaks.
Okay.
And I talked a little bit about this at the CASP meeting, but you can basically solve coordinates or generate data for the 2 different conformations that are there and validate against that. It is very sorry.
Yeah, I would just add, I think exchange relaxation measurements are a source that really that's the main thing that I've been working on, with the goal of creating benchmarks for a real ground truth of proteins that exhibit both fast and slow conformations that we could then actually use to, if we know that, we can evaluate modeling approaches, but then we can also evaluate the heterogeneity that we see in other sources as well. Like we've heard that there's a good amount of heterogeneity in the PDB already, or in cryo. And so
Of course, when you look at exchange, let's say nuclear relaxation or CPMG type experiments, you get information about rates and populations and even chemical shifts, but you don't get coordinates so easily.
You don't, and that's the challenge,
So,
yeah.
and so I think for the modelers, what they want to validate not is how fast it converts or even the populations. They want to just be able to predict the alternative conformational states. But there is a marriage there. Chemical shifts relate to structure. They might be able to get some NOE information or other kind of distance, and PRE experiments could be used in that context. So, but we should keep in mind that a lot of the most beautiful dynamic NMR studies aren't giving us alternative conformations, at least not very directly.
For sure, yeah.
So I think we should wrap up, and already half the people have dropped off, but I want to leave the line open for just a couple of more minutes, 5 more minutes or more, so that people could ask the speakers if there's other questions. Whenever you give a talk, you like to get some feedback, and we had a pretty dense meeting. So I'm going to stop sharing and offer the space for anyone to ask questions for Hannah or Rhiju.
I have to drop off, unfortunately, but thank you, Guy, and the committee for organizing. And Hannah, it's great to see you. See you again soon.
See you
All
too.
right, bye.
So are there any other questions then for Hannah?
It's Monica Roth. I just wondered if you looked at membrane proteins at all as a class of proteins that have
I have not, but actually one of the first papers that demonstrated signal for alternate conformations in evolutionary type modeling was on membrane proteins. So in particular, Debbie Marks is the corresponding author 2012 in Cell. And that was a figure that in Lucy Caldwell is on that paper that really motivated us to think that we could leverage evolutionary information with AlphaFold for alternate conformations. And again, the paper from Jens Meiler's group is also looking at a GPCR and showing that just by injecting noise into AlphaFold that they can sample multiple conformations.
And the Michael Ike paper that we talked about, work that we talked about in the last meeting, was also on membrane proteins, but not so much on ECs.
I have a quick question for Hannah if we have time. I was curious, so with your switch data sets, how much variability do you see in a cluster? So if you look at the two populations, what type of variability and structure do you see? Like, do you see, I don't know, side chain variability? What type of things can you get out within a cluster?
That's a good question. I have not looked at, for instance, side chain variability, and I haven't really calculated that. I am at the moment acting just very qualitatively, just taking, for instance, the top 5 models as ranked by PLDDT to analyze the states.
Hannah, when we studied your paper in our group meeting and we really enjoyed it and thought it was great, one idea we discussed was, rather than clustering nearest neighbors, to use the evolutionary tree to actually do the clustering. And obviously is very related to what you did, and you showed the evolutionary tree. So why would it be better to just cluster on, would there be an advantage to cluster on clade rather than cluster on just sequence similarity?
I think using more knowledge like that, yeah, like for instance, for clade or evolution, ought to create a better clustering. And our goal with this first paper was to develop a method that could get the most information out of a given collection, agnostic of knowing anything else about the system. But I definitely agree.
Does anybody else want to add some questions here? If not, one thing I would encourage people to do is to kind of brainstorm with your friends and identify how we could possibly get data sets or models or data sets that we could validate predictions against. I think it's a very important area. And one of the beauties of CASP is the concept of blind prediction. You don't know the answer, and you're going to test methods where we know that the answer wasn't available. And it's very challenging. It's not just a single crystal structure. It has to be cases like Rhiju talked about, a group got multiple conformations in their study. They're not yet ready to publish that. They're willing to make them available to CASP, maybe crystallography, maybe NMR, maybe FRET even, or cross-linking. So I think where we're going to get these targets isn't so much from these meetings or making them ourselves. It's going to be by letting the community know that this is going on, this activity, and convince them it's worth participating, contributing their targets to CASP. Okay, so we stop there. And please send us, there's an email link that I sent around for this group, and any suggestions or thoughts you have about how we could do this more successfully. Thanks. Thank you, Hannah. That was beautiful. Congratulations.
It's a pleasure to share with everyone here. So thanks so much for the invitation.